Right, hello and welcome to the showcase stage. My name is Dan Fordham and I work in strategic product management here at Oxford Nanopore. In case you aren't aware, ORG.one is a project to support researchers and conservationists to expand genomic resources for endangered species. We provide free of charge flow cells and reagents to Oxford Nanopore users to sequence endangered and critically endangered species. All data is owned by the users and is shared in an open database. You can browse these at the European Nucleotide Archive or through links on our website. These data are often the first genomic sequences for some of the world's most threatened species, and the ORG.one community are using them to inform conservation management, enabling genetic assessments of populations, and to understand inbreeding or assess hybridization or even refine reintroduction efforts. As part of this series, we've now sequenced 15 species live at Oxford Nanopore events, and today is no different. From tigers to butterflies, we endeavour to deliver large amounts of high-quality data to enable genomic assessment of precious samples. And with that in mind, I'd like to introduce Rachel O'Neill, who is Director of the Institute for Systems Genomics and the Centre for Genome Innovation at the University of Connecticut. Rachel, would you like to introduce yourself and highlight some of your research areas?
Sure. Thanks for having me here. I'm Rachel O'Neill. As he said, I direct the Institute for Systems Genomics and our sequencing core, the Center for Genome Innovation, and we work with scientists all across the globe to sequence as many different and unique and exotic species as we possibly can, and that's what brought us to the ORG.one initiative. And we're very excited to be a partner for ORG.one so that we can actually contribute to sequencing endangered species across the globe. So my research is mainly focused on comparative genomics, and I feel like I'm in a very exciting time in biology because we can finally do comparative genomics in the way that we've never actually been able to do before. Because we have this long-read sequencing technology, we can have genome assemblies that are T to T. And because of that, that allows us to do comparative framework type studies across a broad range of species. And we can look for everything from adaptation to different environments, and that's something that we're working on in the Antarctic, for example, to how different phenotypes are actually evolved. And that's something that we're really interested in and very excited about studying. So this is actually one of our groups. So for those of you who don't know, kangaroos and wallabies are endemic to Australia. They are they have a pouch. They give birth to altricial young. That means they're very underdeveloped, and they crawl into the pouch, and that's how their development proceeds. There are 59 different species of kangaroos and wallabies, and you can always think of them like kangaroos are these tall guys, wallaroos are about here, and then you have all the wallabies, and they range all the way down to really tiny. About a third of the different kangaroo wallaby species are part of a group called the rock wallabies, and many of them are endangered, including the species that we're sequencing today, the Proserpine rock wallaby, or Petrogale persephone. They're found in these fragmented habitats, and this is one of the reasons why they're also endangered. They live in rocky outcrops, and they're quite small. They're only about this big. And so the reason I studied them during my PhD because they've undergone rapid chromosome variation. And so every species has a different karyotype than its neighboring species. And we want to know why this happens. This happens in a very small group of mammals. It happens in rock wallabies. It's happened in equids, and it's also happened in gibbons. So it's a really interesting phenomenon that we're interested in. If we go to the next slide. One of the threats to rock wallabies today is that their habitat is being taken over by developments that makes their habitats even more fragmented. But a bigger threat to them is actually feral cats. So many cats that have been introduced to Australia have become feral, and they're quite large. And you can see a cat there that's actually eating a wallaby. Also, cars. Many wallabies get hit by cars or are eaten by eagles. This is actually a wedge-tailed eagle eating a large swamp wallaby. So you can see these eagles can be quite large. Another threat to rock wallabies is that they're opportunistic eaters. They just kind of forage on anything, and there's been a lot of introduced plants into Australia. It is an island continent, and those introduced plants have sort of taken over, and periwinkles is one of them, and while they're really pretty, rock wallabies will eat them, and they're actually poisonous. And so all of these factors are actually contributing to the rapid decline of rock wallaby populations, and in particular, the Proserpine rock wallaby.
Super. Thank you. So this is a fairly interactive session, so we're going to open up for questions. I'll ask a couple of questions first. So I'll go first. Right.
Okay.
Right. The University of Connecticut is a sequencing partner for ORG.one. How do interested participants work with you?
Well, that's a great question. So reach out to us and reach out to ORG.one. We'll work with you to talk to you about the sample that you have, the species, submit the request to ORG.one, and organize when in the queue you would actually want to have that sequenced. And a lot of times it depends on when you have the sample available. And so we're ready to work with anybody that's got a sample that they want to submit.
And do you have any examples of that being successful?
Yes, so we've just recently completed the Puerto Rican parrot genome. We've done quite a number of bird genomes. We've also done some gibbons, as I mentioned earlier. I'm trying to think of some of the other species that we've done. It's a very long list.
Salamander. Yeah. Salamander?
Oh, yeah.
Yeah.
We've done a salamander. We've done some fish. You name it, we've touched it. We've done a lot of plants as well. So you don't have to just stick to animals.
Great. So how important are your relationships with partner organizations with working with species of conservation concern?
We think that's actually one of the most important things. And so one of the things that we've actually done at UConn is we've set up a training program for undergraduates called the Conservation Genomics Biodiversity Training Program. And what we do is we actually take some of the genome sequencing data that's coming out of ORG.one, and we train a cohort of students in genome assembly. And as part of that, they have to work with conservation managers. So there's a clear link between management strategies and the genome sequencing data, because we think one of the biggest divides is actually between conservation managers and genomicists. So how do we actually translate that data? And that's something that this program is really keen to do, but something we think is really important.
I think it's an excellent project, personally. Right. Thank you very much.